All right, so I'll be talking about the enzyme lesson plan that I developed for EMS 521 uh, for my final presentation on November 15th. The materials required for this lesson are relatively few. There is a worksheet. I will have uploaded it. There is a flipped class video. I've uploaded that as well. But for sheer consumables and reagents, all you need are these protease assay plates, which are basically just 1% weight by volume of gelatin, skim milk powder, and agar and water. So hopefully I have water. I didn't include that. You will need food dye to add to the plates as they're being boiled and made, either on a hot plate or in the kitchen on the stovetop. You got to melt the agar and the gelatin into the water just so it sets. It's the same concept as agar plates. And we find that blue, green, or red food dye works quite well. In the future, I would actually add 4 drops of blue and 4 drops of red to make a darker purple, which would increase the contrast on your plates. Then you need the Petri dishes, and we used 100 by 15 millimeter Petri dishes, and that worked really well. For fruits that we used, apple, banana, mango, orange, pineapple, and tomato worked the best. I would recommend sticking with these. These were the easiest to homogenize or crush with a mortar and pestle. If you were to expand to other fruits, I would recommend using a blender or food processor. And basically 5 grams of fruit, you add that to 5 milliliters of a saline buffer. I chose to use a phosphate buffered saline at pH 7.4. Any sort of physiological saline, like Ringer solution, would probably work. Just make sure the pH is around 7. So a lot of these proteases are going to want to be close to neutral, maybe even slightly acidic. So that's technically all you'll need. Everything else I will have uploaded, the flip video, the worksheet, and then this video itself kind of walking through all the stages. The standards that are addressed by this lesson plan, the primary standard is Bio 4.13. Explain how enzymes act as catalysts for biological reactions. The flip video will have handled that, and then the lab work during the class will kind of reinforce what's taught about enzymes. Then the secondary standards, we will be reinforcing scientific inquiry and experimentation. Part of this lesson plan is an argumentation module. So students will generate data, then I will add in extra data sets, and then they will look to make claims and arguments around these data sets. The lesson plan, and feel free to skip to the next slide in about 15 seconds, but this is just how I wrote it down for my understanding. The flip video is going to end with a question around amylases. So the flip video talks about enzymes, and I end that with a case study on amylase activity. And then I end with a question: do cats have amylase activity in their saliva? The class will start with a discussion around amylase activity in cat saliva. The prompt then, because we're already talking about mouse and enzymes in there, we'll actually talk about what happens when proteases get into our mouths. And a lovely, horrifying example is pineapple. Pineapples have a boatload of proteases in them. And we'll talk about what happens when you have too much proteases in your mouth at one time. It's a bit gory. And then this will kind of lead to the question, we're all kind of teaching scientists and we want them to become better scientists, so we should be asking questions. So why do pineapples have all this protease activity? Is this typical in fruit? Do other fruits have this activity? These are all questions that should be answered, and these are questions we're going to address in this lesson plan. And then finally, we'll do kind of experimentation around looking at protease activity in fruits. We'll do the argumentation module, basically looking at the data, making claims around that, answering a question that actually is not answered in the literature. Why do pineapples have a lot of proteases? We'll get a consensus between the different groups, so which claim or argument do we support the best? And then finally, we'll talk about next steps. What experiments should we perform to kind of answer claims we have? If you're more comfortable with the 5E lesson plan system, it's a lot more streamlined, so I probably should have started with this. The engaged, we have the flip video, the discussion around cats and amylases, and then the perils of pineapple. To explore, we're going to do experimentation to determine protease levels in different fruits. The explain and elaborate form the core of the argumentation session of this lesson plan. And then finally, we'll evaluate and check for understanding by asking them to continue. What are your next steps? So if the foundation's there, they will be able to generate at least an idea of what experiments we should continue with. So for today, I'll run through the lesson like this. You will see the slides that were used. You'll see the experimental setup. You'll see the results. You'll see the arguments and the consensus, and then you'll see the next steps. There is going to be no video of this actually occurring. This has been demoed with my colleagues, but not in a public setting that we felt comfortable videotaping. So where I work, we are actually scoping this out, and we will take this to classrooms in 2019 because there was quite a bit of excitement and interest in this demonstration and lesson plan. Particularly as it fits one of the North Carolina state standards. So if you see this, if you like what we're seeing, if you don't feel comfortable replicating yourself, reach out to me, and either I can come into the class and do this with you, or I probably can find 3 or 4 volunteers who were actually super excited by this lesson plan and demonstration. So yeah, everyone who did that basically is stealing that for their children's science fair projects, which I have mixed feelings about. But first, go and check out my flip video first. It's about 10 to 12 minutes. It will cover the enzyme portion, and then the case study around amylase activity in human saliva, and then end with a question around cat saliva. If you've seen it, we'll kind of continue. Basically, to reiterate what was shown, I basically calculated that the saliva in my mouth is capable of degrading 2 saltine crackers in 1 hour, which is not as much amylase activity as I was hoping for, because clearly I wanted to sell my spit to convert starch to glucose for these bioreactors who are going to use that glucose to make ethanol. So I'm not going to make money from my spit anytime soon. I did end with a question around whether you expect my cat to have amylase activity in his saliva. And once again, aside from me getting saliva and scratching up my arm, what do I expect to find? And when I actually talked to my colleagues about this, they almost all got it correct that cats are carnivores, so there's not going to be amylase activity in the saliva. What's the point? They're not encountering starch, so not eating plants. And if your students have issues with this, the easy prompt is, well, are cats omnivores, carnivores, or herbivores? Most people know that cats are carnivores, or I'm hoping most people know. They even say, well, lions are closely related to cats. Lions, are they carnivores? Yes. And so because they're carnivores, they will never encounter plant starch, so there's no reason for there to be amylase activity in their spit. And the citation's down there if you're curious. This leads into, well, we have amylase activity in our mouths. There's another class of enzyme called proteases. They're not that heavily expressed in our saliva because they're very good at degrading proteins. Unfortunately, our cells are composed of a lot of proteins as well, and we don't really want to degrade our cells. So the perils of pineapple's an amazing fruit, but if you eat too much of it, it will actually cause your tongue, your cheeks, and your gums to bleed, as well as cause lesions on your lips and inside your gums. An amazing fruit, by the way, but you should never eat too much, and you should never eat it when your tongue starts to swell, because there is actually cellular damage. And so I post this up because it is quite a lovely indication that pineapples are vaguely cytotoxic. They will lyse cells because of all the proteases within them. I would not recommend showing this video in class. I think the picture itself is beautiful, but the video is got strong language and quite a bit of gore, and it's not reinforcing a lot of good behaviors. But it was a nice indication that these proteases are if you get too much in the wrong area, they can actually do a little bit of damage. And another indication that's a little bit more family friendly is we use pineapple to tenderize meat very frequently. And meat is composed of proteins, as all animals are. So you can imagine pineapple is degrading these proteins, making it a little softer, a little easier to digest, modifying the flavor slightly. And you can imagine, well, if it does this to meat, what could it do to me, who's just a big bag of meat? So that's kind of probably a safer segue for you. And everyone in your class hopefully should have at some point seen the pineapple in gelatin experiment, and they should be familiar with the protease concepts around that. But in case they're not, pineapples are composed of a compound that's called bromelain. Bromelain. I call it bromelain. I've heard bromelain. It's basically composed of several different proteases. Most of them are cysteine proteases. How they work is they find proteins, find the cysteines, and using water, they will actually degrade them down into polypeptides, which are just shorter proteins. And eventually these polypeptides will become smaller and smaller and smaller until they're just little peptides or down to the amino acid components. So they're basically degrading the protein into smaller usable subunits to make more protein if you want, or to digest and metabolize. So the question that I always asked whenever I saw this kind of science fair project, either when I was growing up or recently as I do more and more kind of judging at these things, as well, why do pineapples have all these proteases? Is there a way to quantify it and then compare it to other fruits? Is there a way to kind of bring this more mainstream so it can be a nice demonstration? And the answer is like, yes, we can sort all that out. So I took a bunch of fruit, homogenized 5 mils of that, 5 grams of that in 5 mils of buffer, and then I applied them to the plates we talked about earlier. And so there are 3 millimeter holes I cut into the agar, and then I added 10 microliters of my fruit extract in there. And because the plates have skim milk in them, which is composed of milk protein or casein, which is cloudy, as the proteases degrade this milk protein or casein, the hole you will see will get bigger and bigger and bigger as the milk protein is digested by the proteases. And you can start to see some of that happening here. And you can see this in real time. The reaction is relatively quick. It can take up to an hour, but you can observe it. When we compared all our different fruits, and once again, this reaction can take up to an hour, what I would recommend for classes is you take it to this point. That's generally a class period or close to it. Put it aside overnight, and then the next day put it in the refrigerator to stop the reaction. And then the next time that class comes in, you continue with the data gathering and the argumentation stage. So at this point, our negative controls here, we have no fruit in here. There's been no expansion of the circle, so there's no degradation of the protein. Apple, you can see a little halo around that. So yeah, there's a little bit of proteases in there. Orange, slightly bigger, so a little bit more protease activity there. Tomato, just a little. Banana, a very tiny amount. Mango, a good chunk, a nice circle around there. So yeah, there's a lot of proteases there. And then finally, we have pineapple. Nice big circle here. So we know there's a lot of proteases there. So what you would have your students do when this is all done is measure the diameter of these halos or clearance zones. And that gives you an indication of how much protease was in that fruit extract. And here's the data. It's very simple to calculate. You basically take the area of the cleared circle and then just subtract the area of the initial 3 mm hole. And to calculate the area of a circle is pi r squared. Most students in high school should know that. But if they do require help, just prompt them, put the equation up on the either whiteboard, PowerPoint, or in the handout I'm handing out. So we get actually a really nice kind of range of protease activity from these different fruits. We do find that pineapple is the highest, closely by mango though, and then intermediate levels in orange, tomato, apple, and then next to none in banana. So it does seem that pineapple does have the highest protease activity of all the fruits I tested. Mango's close though. So this was the data gathering experimentation stage. We did have a negative control. Our pineapple we expected to work, so that was our positive control. And then we had a wide range of different fruits to kind of test and look for different levels of proteases. So it was a nice experiment. Every group of kids can run all of the fruits. That way you can compile the data and get a nice running average. What I then did, to complicate things, they thought they were done when I ran this through with my colleagues, is I added data columns. So here is the data that they generated, and then I added in data on the fruits, including time from flower to fruit. So this is just development time to fruit. How long these specific kinds of fruits have been domesticated for, and there is a lovely range there. Where it was originally domesticated, and then the rainfall where it's typically cultivated. So whether they're experiencing a lot of rain or very little amounts of rain. And so for this, and this is in the worksheet that I've kind of uploaded, you'll kind of run through the argumentation stages, phases, where they're going to generate a research question, which this is mainly already done. Why are there different levels of proteases in fruits? The claim, and they're going to be comparing their data to the data I present to them and drawing conclusions from that. They will then show their evidence. This is typically a sentence or two because it's pretty straightforward. And then their rationale for why they think their claim is true and that the evidence supports it. So, and my groups did it slightly different. They actually gave me the biological reasoning why we would expect this to happen. And I really like that, and we'll show that in a minute. So one group, their research question, why do some fruits have greater protease activity? Their claim was that fruits that take longer to develop have higher protease activity. Their evidence was based upon tomatoes, apples, and bananas have the shortest development time, so they have the least amount of protease activity. And then the rationale, and these are scientists, so it was a biological rationale. It was quite interesting. The rationale was that the longer a fruit takes to develop, the more the plant will want to put in defensive compounds like proteases to prevent people like me from eating a ton of pineapple or mango. And I really like that because that tied in kind of the initial proteases chewing up the gentleman's mouths earlier. And so I like that the evidence, even though it was anecdotal, was used later on with the evidence that I provided them. The second group, a very similar research question. Their claim, fruits of plants that get less water have more protease activity. Their evidence, pineapples and mangoes have the least amount of rainfall, or where grown, yet the highest protease activity. And the rationale, once again, was very similar because there's not a lot of water. Fruits are going to take a lot of resources to make, and they're going to take a larger share of the water available to the plant. And as a result, they're going to want to protect it a little bit more. So, yeah, they basically took that in as well. And finally, the last group, very similar research question, and their claim was a little bit more complex. Fruits of plants in long, dry growing seasons have greater protease levels. Basically, they put the claims in the two previous groups together and made a more encompassing claim, which I really liked. And then the evidence was the same as the other two groups, but compiled. And then the rationale was, once again, it was around protecting themselves with these different proteases. So if we were to go back and look at the data, if you were to take this data and actually graph it out, the protease activity does correlate relatively well to development time and rainfall. So the longer it takes for a fruit to develop, the more likely it is to have higher protease activity. The less rain a plant is exposed to, the greater the likelihood it has higher protease activity. So an inverse correlation with rainfall. So the data set's all worked out. All of my colleagues who ran through it really liked it. They liked the argumentation session. They liked arguing with each other because they're scientists. And we came up with a consensus, and that was the last group. So, and at the end, I did ask them, we saw this. We got pretty nice correlations. How would we test this experimentally in a lab? What are the next stages? And I got a lot of nice suggestions from the scientists and one non-scientist I had run through this, giving one type of plant different amounts of water, seeing how that impacted the protease content. Knocking out, these are scientists, so we do genetics, knocking out the proteases you would expect to find in the fruit tissue and seeing what the impact was on colonization by insects or pathogens, things like that. So that was neat. So there's lots of different things you can do for experimentation to support the arguments we're making based on correlation. So finally, my conclusions, this actually was a relatively easy lab, and we made it with readily available materials you could find at Harris Teeter or Food Line or Walmart. And so it is easy to modify for different grade levels. You would just add or subtract information, or add or subtract fruits. Younger kids or kids that need a little bit more help, mainly just focus on the experimentation. I think that's reasonable. I think the argumentation is probably going to be more for high school students. It was nice because it allowed for visualization of enzymatic activity, which I've actually not seen a lot of demos that do that. So you can actually see the circles get bigger and bigger in their plates. I thought that was really nice for me. And finally, this was kind of a do-over for that horrible pineapple experiment that everybody does for the science fair project. So it basically vindicates and validates that experiment by assigning numbers and allowing us to ask the question, well, why does it have a high level of protease activity? And how can we kind of generate hypotheses around that? How can we test that? And finally, what I really liked about this demonstration, I did a lot of reading around, why do pineapples have a lot of proteases? Was it something that was bred in? Is it just some sort of oddity because of some sort of predator or pathogen? And actually, no one has an answer for that. So we were actually asking questions in this demo that there is no ready answer in the scientific literature, which really excites me because these are kids kind of learning something brand new that no one has any idea about, and then forming hypotheses and claims that's new. It's not in the literature. It's not something that's been heavily written about. It's something so ubiquitous in our science fairs and demonstrations. The pineapple degrades gelatin. Everyone knows that. But no one asks, well, why do pineapples have all these proteases then? Why are they doing this? This is just weird. They're not exposed to gelatin in the wild. And so I think it's really cool because then you can explain to students, well, actually, no one knows why. We probably have a better idea than most people right now because we looked at it. So that really excited me. So that's my lesson plan. Sorry I don't have video, but hopefully with this video, you'll be able to either recreate it in your classroom, or if you're interested, please reach out to me and we can kind of collaborate on getting it into your classroom. It is a very easy demonstration. I like the incorporation of the argumentation module. I like the flipped video concept because a lot of the teaching is happening before the class and you can really focus on the demonstration and the inquiry-based science. So yeah, Yeah, thank you for listening. I really enjoyed kind of putting this lesson plan together and kind of executing with my colleagues, and they really enjoyed it. So thank you.